Morning everyone. Today I am going to teach you about LabXchange. It is a free and open source website which has plethora of contents and tools to teach science. It is especially for teaching of science. And the name is labexchange.organization, labexchange.org. So it has lot of content, not only on science. It is mainly for the science, but it has content on various other subjects also. So there are two modes to log in or to sign up on LabXchange and to log in. The first one is educator mode and the second one is learner mode. So in the educator mode, you get lot of features to modify and edit the content and to create your own content also. But in the learner mode, the features are limited. This is basically the learner mode is for the students and the educator mode is for the teachers or educators. So I have already signed up and logged in as educator. So I will explain all the features of the educator mode. First, we will see the home page of this LabXchange. So this is the home page of LabXchange, and we can see that it has educational resources and contents from over 150 leading organizations. And if we click on this, then we can see all the list of all the contributors. So the main sponsor and contributor of LabXchange, which sponsored this LabXchange, is Amgen Foundation. Amgen Foundation is a foundation which is set up by Amgen. Amgen is a biopharmaceutical and biotechnological uh, company, and LabXchange receives its fund from Amgen Foundation. And the strategic allies is the UNESCO. It is also supported by UNESCO, and the content collaborators. You can see this list. So we can see this list. It is Amgen Foundation, and then we can see lot of BioBuilder, biology simulations, brain facts, cell mentor, Chronos, and Duvool. Duvool is from India, and Eureka, GeoGebra, Harvard, lot of Harvard here, and HHMI, Biointeractive, Iris, I'm a scientist, iBiology, Khan Academy, LabXchange, Lifeology, Meristem, the Nobel Prize, Professor Dave. There are more than 150 contributors for this. FAT is also there, Concord Consortium. So this is the list of all the collaborators, all the contributors. Then if we go back to the home page, then what else is there? Uh, the process is all you can see, you can explore all the content for free. You can also sign up if you want to become an educator, or you are a student, then you can also sign up. It is free, and then you can share also. And it is the Amgen Foundation. About Amgen Foundation, so it has contributed more than $400 million to LabXchange till today. So we can first go to the explore section to see what is there. So here we can see featured subjects. If we click on the featured subject, then it is a list of all the subject on which content is available. So we can see biological sciences, chemistry, global health, health science, science and society, scientific progress, prepare for college, prepare for graduate school, prepare for careers, professional development. So on all these subjects or topics, content is available. If we click on biological sciences, we can see what is available. So there is a lot of content available on biological sciences. We can click on see more to see what else is there. Then on chemistry, there is a lot of content again. Then we can click on global health to see what is there. Factors that affect air quality, COVID-19 vaccine doses administered. We can click on see more to see what is there. And then health sciences, what is cancer, and then HPV, adaptive immunity, and then on science and society. So this is the content on science and society, then scientific progress. Then we can see biosafety levels, and then we can see on prepare on college, the type of videos for career, for advice of the career, or for like how to prepare for university exam, how to study. We can click on to prepare for graduate school, like pursuing a PhD, and then prepare for careers. And then so here meet a scientist, IGI scientist, and then career day, choosing a career, and then we can go to professional development. So here we have 5 teaching ideas for a new semester. Science exams don't have to be demoralizing. So just science. So a lot of content is there. And then if we get back to content type, we can see what is there. Then we can see there are a lot of content on LabXchange, like clusters. Clusters are a lot of different type of content. They are clustered together to make complete package. So it is called cluster. And then there are pathways. Pathways are serially arranged contents. Different type of contents are serially arranged to make a complete chapter or a lesson. So these are pathways. And then there are interactives. These are simulations with which we can interact, we can modify, we can edit, we can do lot of, we can change lot of variables. And then there are simulations. These are like games, we can play with them to understand a concept. And then there are videos. Then there are narratives, people who teach something, teach a concept, who talk about it. Then there are case studies. Case studies are the things we can like see, like pathway. These are all our clusters. And then we can see pathways. So in pathways, there are various items that you can see, 13 items, 9 items, coronavirus 9 items. So in pathways, there are a lot of items which are arranged one after the other. There can be videos, there can be text to make a complete lesson. Like structure, like we can see, there are 13 contentancies. So these are text, then question set, then text, then question set, then question set. In the interactives, these are very interactive videos, like we can see here, or we can see more interactives here. Interactive human body, COVID-19 vaccine development, transcription. So there are a lot of interactives. 776 interactives are there. Like we can open this structure of DNA. So here it is written, scroll down. So here it is explained what is DNA. DNA or deoxyribonucleic acid. It is a polymer, basically polynucleotide chain, and then there is a backbone of sugar and phosphate, and nitrogenous bases protrudes inside, and they are connected with each other by the hydrogen bonds. So this is the explanation of DNA, the 5 terminal and the 3 terminal. At the 3 terminal, there is a hydroxyl group, free hydroxyl group at the third carbon, and at the 5 terminal, there is the position of the carbon. There is a phosphate group. So it is called fifth terminal, and it is called 3 terminal. So we can see what else is there. These are the nitrogenous bases, adenine, thymine, guanine, cytosine, and here it is the color code of these. These basically the basic sugar phosphate backbone remains the same in every DNA. It is always the same. It is these combination of these nitrogenous bases that make DNA unique. So we have this covalent bond. Uh, we have this phosphodiester bond between one nucleotide and another nucleotide. We have a phosphodiester bond, and then we have a glycosidic bond between nitrogenous base and this sugar. This is the pentose sugar. For DNA, it is deoxyribose sugar, and for RNA, it is a ribose sugar. And these are hydrogen bonds. So between A and T, there are double hydrogen bonds, and between G and Z, there are triple hydrogen bonds. These are stronger. These are a little bit weaker. And this is a sugar phosphate. This is a sugar and this is a phosphate. So they are sugar phosphate backbone, and these are nitrogen bases which protrude inside. These make DNA unique. These sugar and phosphate are same for every DNA. These are negatively charged, phosphate have a negative charge, and these are same sugar phosphate for every DNA. These are unique. These inside nitrogenous bases, which we can see on orange, blue, yellow, green. So these are biointeractives. These are interactives, and then we have many other things also. So we have simulation. Simulation is like, first we will give the overview. Simulation is like game which you can play to understand a concept, and then there are videos, lot of different videos, like video, like we have video of glycolysis, then secretory pathway, then videos of people. You can see, click on see more to see a lot of different videos are there, and then there are narratives, people who talk about a particular topic, like no place like home, and change and uncertainty influence opportunities in our life. Then we have case studies, experiments about like this is a green fluorescent protein of fluorescein, and then we have phages about bacteriophages. Phages can be more effective and specific than antibiotics in combating bacterial infections. And then we have documents, the texts like engineering, golden yeast, how does the brain work? And then we have question sets. These are basically the problem sets, question sets to under for assessment purpose. And then we have teaching guides. How to This is basically for the teacher how to teach, like skin color, human evolution, bloom, taxonomy, coral reef, and global warming. And then we have texts for documents on the things like magic mushroom and amygdala, infographic, fluorescence, Rosalind Franklin. And then we have images, lot of different images, protein synthesis, ribosome structure. And then we have audios about Alzheimer, then different type of audios. And then we can just go to content sources. We can see who have contributed for this. This is a long list, very long list of all the content creators, community favorites, the videos which have received maximum likes or views, and then trending topics right now. And then we can go again to this, explore, and then subjects. So we can see that for every subject there is middle school level, then high school level, then advanced level. In the middle school level you will understand the most basic fundamentals, then the high school level, and then the advanced level like genome editing, cell cycle, DNA replication, biotechnology, chemistry of life. And in the middle school you will understand about the basics like what is DNA, ecology, photosynthesis, evolution, introduction to cells. So here you will study introduction to cells, and advanced you will study about cellular energetics, how cell obtain energy, what happens to with the energy production, cell cycle. So in the chemistry, the same for chemistry, the atom, properties of water, matter and change. And here you will study Le-Chatelier's principle, rates of reaction, chemical kinetics, nuclear chemistry. And for physics also in the middle school, then high school, then advanced, and earth and space. So here you can see that there are four subjects, but these are not only the subjects. There are more content on this. We can go into the library to see what content is there, and then we can go to the subject area. So here you can see there are a lot of content is available on LabXchange. Biological sciences, for which more maximum content is there, 14,000, more than 14,000. Health sciences, then physics, then chemistry, earth and space science, science and society, scientific progress, global health, prepare for careers, educator skills, environmental science, data science, economics, learner support, mathematics. And then you can go to background knowledge. So for 12,000, more than 12,000 content, or about 13,000 content, you don't need any background level knowledge. This is the most basic content. And then for some like 8,000 you need some background knowledge, and for 1,000, like 1,300 content type of content, you need extensive background knowledge. And then we have for video, you can sort or filter for video duration. Then source, you can see all the contributors here. You can see the name, OpenStack has contributed the most. Then LabXchange itself, Meristem, Futurum Careers, NASA, NIH, Harvard, Khan Academy. So you can see all these. Then popular tags, you can use these popular tags to search for a particular content like DNA, climate change, earth science, environmental science, high school, botany. So you can use these popular tags. And then language. So the content is available in lot of language. This makes LabXchange very unique and very special because it is completely free and available in lot of different languages. So you can see all the list of the languages for which content is available, and then you can go to the sources which have contributed for this. So it is showing 170 sources, CDC, Harvard, tag. So these I we have seen all this: BioBuilder, Harvard, Amgen Foundation. And then you can filter them by their country, by the country, United States and UK, and then the language. So then we will see about dashboard. So I'm an educator itself, but I have not created anything. So I'm an educator. So it is showing create content. I can create content. So there are different type of content which I can create, like question set, questions. These are basically for assessment problems, audio, case studies, document, images, narratives, pathway, teaching guide, text, video. I can create anything on this. And then favorites, the things which I have liked, uh, and then classes I teach. So I can create a class also. Like we can name a class, we can choose a subject, the learners, so maximum it goes to 300 learners, and then we can give a description of the class, we can upload a preview image, and then we can add co-educators or teachers for the class. So it is just like Google Classroom. It is much more than Google Classroom. We can do various things here and it is completely free. And we can create assignments, we can create a lot of different videos, we can share it, and class I'm in, and then messages which I receive. Then we can go to community. In the community, there is a discussion page. So in the discussion page, like there is a community of science communication, education and careers, teaching of science, learning science. So this is a community. You can join any community and then you can share knowledge about these things. LabXchange announcement and support LabXchange community. You will get information about this. You can discuss about with the peers. And then there is mentees. I don't have any mentees yet, and then there are mentors. So I don't have any mentor yet. You can search for mentor here, and these are the people who are mentors on LabXchange. So you can connect with them and you can make them your mentor. The library section to see what is there. So we can also go to subjects, and we can do some like teach some concept, or we can do something here. So we can explore this. First, we can go to, like, we can take any topic from physics. So here we can take any topic like gas molecules in motion, projectile motion, Faraday's law, greenhouse effect, forces, and so we can use this circuit construction kit. It is by fact. Here you can see that it is a simulation. For every content there is this box for which you can see that what is it. So it is a simulation. Language is English, subject physics, background knowledge, yes, you need some background knowledge. Favorites 25, views 5,900, remixes 7. So people have edited it and uploaded. So 6 are private and 1 is public. So you can yourself edit it or modify it if you are an educator. So these are remixes, and we can start the simulation here. So it is a simulation by fact. So first we can take a light bulb here, electric bulb. We can connect it with some wire. We can connect an emitter here. We'll take some more wire. So more wire, we can connect a battery, one more battery, and then some wire, and then a resistor, then some wire, then we can connect a switch, some more wire. We can just do this, and then we can have a fuse here, we can connect it here, then some wire, then we can use something like, let's take some more wire and connect with it. So now this circuit is complete, but it is open. We will close the switch and the bulb is glowing right now, you can see. We can take a voltmeter here to check the voltage, connect it here at the battery. Yes, so our one cell is 9 volt, so we can see the rating here, it is 18 volts. The voltage of two cells are combined, gets combined in series, and it is 18 volts. And if we increase the voltage, then if we remember the Ohm's law, then Ohm's law says that V is proportional to I, that voltage is proportional to current. That is, in a given circuit, if the external factors or conditions are not changing, like if we are not changing the resistance, if we are not changing the temperature, then voltage is proportional to the current. If we increase the voltage, then current will increase in the circuit. So we will see the Ohm's law here. We will see the effect of Ohm's law. So we are going to increase the voltage. It is right now it is 9 volts, so we are increasing the voltage. It is right now it is total combined voltage is 18.5. We are increasing it, and we can see the effect on here in the ammeter. It is showing amps in the ampere, the current. So we can see the effect on current. We are increasing the voltage here. So we are increasing, and you can see that when we are increasing the voltage, then the current in the circuit is also increasing. Like now it is more than 1 ampere, and we are continuously increasing, and the current, as current is increasing in the circuit, then this bulb is also becoming more brighter, and the flow of electrons in the circuit, we can see it is becoming much more vigorous, and the bulb in there, these rays, you can see these are spreading out, and we can continue to increase the voltage, and you can see that the now it is much more brighter. We can delete this fuse, and we can just elongate this. So we have confirmed the Ohm's law, that we have increasing the voltage, then current in the circuit also increases. And so what we can do now is, we know V is equal to IR. So R is equal to V upon I. That is, if we increase the resistance in the circuit, then the current will decrease in the circuit. So we are going to increase the resistance on the circuit, the voltage will remain the same, but the current in the circuit will decrease. So we are increasing the resistance. You can see the color codes are changing. These color codes actually, color bars actually show the resistance of the resistor. This is a method to represent the resistance. So you can see it is changing. We are increasing the resistance in the circuit. The electrons are not easily able to flow, and current is basically the amount of the electron that is passing through a cross-sectional area of a wire or a conductor per second, which is crossing this cross-sectional area. So less electrons are moving right now. It is, color codes are changing, the resistance is increasing, and we can see that current is decreasing in the circuit. So according to Ohm's law, if you have to make, if you are increasing the resistance, then you have to increase the voltage to maintain the same level of current. So I am increasing the resistance, but I am not increasing the voltage. That's why the current is decreasing in the circuit. Now, to maintain the same amount of current, or to increase the current, I have to increase the voltage. So now if I will increase the voltage, then the current will also increase. So if you are increasing the resistance, then you have to increase the voltage also, because resistance is resisting the flow of electrons. Resistance is decreasing the flow of electrons. Its name is resistance. So it will provide resistance to the flow of electrons, and the battery or a cell does the opposite thing. It creates an EMF, electron motive force, so or electromotive force. So it is the force that drives the electron, so it will push the electron in the circuit. So basically what is voltage? Voltage is the amount of energy which is consumed to drive the electrons. So if you are increasing the resistance in the circuit, then you have to increase the voltage also, because that voltage will drive the electron. If I will increase the voltage, then the current will again start to increase. So that is the Ohm's law. And we can see some more simulations. We can go to the back to the library, and then subject area, and then we can explore chemistry this time. And we can choose any topic of chemistry from here. Like there is pH scale, diffusion across a semi-permeable membrane, and then we have hydrogen bonds. So we can choose this hydrogen bonds. So hydrogen bonds, we can see that it has received 13,000 views, favorites 101, remixes 82, 5 are publicly available. You can click to see this remixes. So people have created these remixes with hydrogen bonds. People have used the that simulation in these pathways anywhere, and then you can go back and you can see there are 82 remixes and licensed LabXchange. So can you start the simulation? And hydrogen bonds are basically, these are dipolar interactions between the molecules. It can be intramolecular or intermolecular. And the special thing about hydrogen bonds is that there is a hydrogen atom in a molecule, and there is a highly electronegative atom, the atom which has very high electronegativity and electron affinity, like nitrogen, oxygen, or fluorine, and hydrogen is connected with it. So the hydrogen, because it has the tendency to give the electron, it is highly electropositive. So it usually gives its electron partially, not completely gives its electron, but partially only. So it gets a positive charge. So water is a highly polar molecule, and water has a lot of hydrogen bonding in it, which means water is H2O. It has every molecule of water has two hydrogen atoms in it and one oxygen atom in it. So oxygen is highly electron loving. Oxygen has high electron affinity. So oxygen pulls the electron to itself. Electron attracts the electron to itself. Hydrogen gets a partial positive charge, and oxygen gets a partial negative charge. So there is a double partial negative charge on oxygen because there are two hydrogens, and hydrogens have partial positive charge. So if we click on this show partial charges, then you can see that oxygen has a negative charge and hydrogen has a positive charge, and it has partial positive, double partial negative charge, and there are two hydrogens. These white are hydrogen, this red is oxygen bigger. And this red gets attracted by the white because of opposite charges. This is negatively charged, this is positively charged, and this white will attract to this. And this is the hydrogen bonding. From this to this white to this red, or hydrogen to oxygen, this red to this white, oxygen to hydrogen. So these are hydrogen bonding. This white will attract this red, this white will attract this red will attract this white will attract this red, and this is the hydrogen bonding. So we can start this simulation to see the effect of temperature on the hydrogen bonding. First, we will see what happens in the ice, in the case of ice. Why ice is solid? Why is ice in the solid form, exists in the solid form? Why water exists in the liquid form, and why there is steam or water vapor in the gas form? Both are pure form of water. Both are the physical forms of water, but existing in three different physical conditions, three different states. So why does this happen? So we will start this simulation. We can see that if we cool down it, if we refrigerate it, if we make a ice, then what happens in the case of ice? So we have reduced the temperature, and now we can see that the water molecules, due to hydrogen bonding, they are coming to each other, they are attracting to each other. We can cool it more, and then it will form associations. They will associate with each other. We can see the formation of these hydrogen bonds. These are the hydrogen bonds. We can see these lines, and we can see how they are combining with each other to minimize their energy. So there is very less movement. We can cool it more. Now it has almost become stable. So this is how ice is formed. Now we can heat it. We can give it heat energy and see what happens. So as we are heating, we can see it is vibrating more. We will heat it more, and we can see that now they are showing movement. We can heat it more, and now they are vibrating vigorously. We can give more heat energy. Now the hydrogen bonds are going to break very soon. Now hydrogen bonds are break. They are like they are very weak. They are just associating and dissociating. We can give more heat energy to them, and we can see the hydrogen bonds have are very weak now. They are coming into the basically they are dissociating, and we can say that the water is at very high temperature, and we can, like, there is no hydrogen bond. We can see now the temperature is so high that there is no hydrogen bond. So this is steam or gaseous form of water. The molecules are separate with each other. So this is what happens with the hydrogen bonding of water with the temperature, effect of temperature.
So that's all for this video. We will meet again in another video to explore some more content on LabXchange. Thank you.